got a pretty good background in muscular physiology. Picture on the first page, obviously, really nothing theme-wise that I'm talking about. Just, you can see the muscles. I like to tell students that used to be me, but obviously you guys know it's not. Now, in that picture, the guy flexing his guns, he is showing you the definition of what we would call his skeletal muscles. But as you guys have been made aware for a while now, there are two other very important types of muscle in our body. What are those other two types? Cardiac muscle that's found in our heart. Smooth muscle that's found in places like our blood vessels, our stomach, our intestines, the uterus, the bladder. On and on and on and on and on I could go. Now, those three different types of muscles share some properties, and we'll kind of talk about and compare them here in a couple slides. All three of them contribute to homeostasis, either by sustaining a contraction, so like holding a contraction, or in many cases alternating between contractions and relaxation. There are four principal or primary types of functions that our three muscles provide. The first two listed here, producing body movements and stabilizing body positions, relate strictly to skeletal muscle. Okay? Cardiac muscle does not help us move our body, nor does it help with stabilizing positions. Same thing for smooth muscle, it's not involved with that at all. The third function there, moving substances within the body, relates to those other two types, cardiac and smooth, which is moving substances like blood from the heart, out of the heart by way of the heart muscle pumping. Substances in the digestive tract, thanks to smooth muscle, are able to, move around and be pushed forward. We've also got sphincters that help hold the food in the certain organs and only allow them out in certain kind of increments. Sperm and egg cells or oocytes are moved through the reproductive system passages, urine through the urinary system, lymph through the lymphatic system, so on and so forth. I could go into much more greater detail. But those all are examples of how smooth muscle works. And then the first bullet there under the moving of substances obviously relates to cardiac muscle. Fourth function really relates to all three types, but predominantly skeletal muscle, since it's obviously the most prominent and we have the most of it, generating heat. And this is something we've talked about since the very first lecture of the semester, when we discussed feedback and how when we get hot, we sweat. When we get cold, we shiver. Shivering is a way that we increase our heat production, and contracting muscles produce a lot of heat, which is why we shiver. It also explains why when you exercise and you're physically active, you get hot and sweaty. The term thermogenesis is a term that literally translates to mean heat producing, and something that our muscles definitely do a lot of. Although bones provide leverage and form the framework of our body, they cannot move body parts by themselves. Motion results from alternating contractions and relaxations of, and I really could put in here, skeletal muscles that make up about 40 to 50% of total adult body weight. And I include this slide mainly because what we'll be talking about in this lecture predominantly is related to skeletal muscle. Obviously that statistic there, 40 to 50%, is dependent upon your body makeup. Whether you are, a fitness guru like the guy that was on that first page, you probably are going to have a lot more, or if you're a couch potato, you're going to have a lot less. Regardless, your muscular strength reflects the primary function of muscle, the transformation of chemical energy that we get. Ultimately, we can trace back to the sun, but more so from food that we convert in our body to ATP, which then we use for mechanical energy to generate force, perform work, and produce movement. Okay, what I'm going to do for the next 3 slides is talk about the 3 types of muscle, their characteristics, how they're similar, how they're different, which is something I do in anatomy. So for those of you guys that are in anatomy, this should be a review for you. Skeletal muscle is named because it's what it's attached to, the bones, our skeleton, and moves them in many cases. It is described as being striated, which is a description that we'll talk about in this lecture that relates to why this picture looks kind of fuzzy or blurry. Striations are kind of stripes that our muscle tissue has, skeletal muscle tissue has, where there are these alternating light and dark bands, what we call striations. When you look at it through a microscope, which is what this picture is showing you, what it looks like 400 times with the naked eye can see. We've talked about this before, but skeletal muscle tissue works mainly in a voluntary manner, and that's because it's regulated by that somatic nervous system that we've talked a lot about. Now, some muscle is controlled more subconsciously, like our postural muscles that run up and down our spine and neck. The diaphragm is a great example of one that is predominantly controlled subconsciously, but we can override that action when we want to stop breathing, because that's what your diaphragm is responsible for controlling. Why we would ever want to stop breathing, it sounds kind of stupid to students when I ask that question, but a lot of times students figure it out. It's important, like when you take a shower, jump in a pool, that kind of stuff. You don't want to inhale the fluid into your lungs, because that is going to cause you to drown. Skeletal muscle attaches to bone predominantly, but also in places to skin, connective tissue called fascia, as well as other muscles. It is multinucleated, and we'll talk about why that is, but that means it has multiple nuclei per cell, and the nuclei are typically pressed up against the plasma membrane, which is unique because the other two types, as we'll see here on the next page, are uninucleated. Typically, most cells in the body are uninucleated, meaning they have one nucleus per cell. Cardiac muscle tissue is like that, as is smooth muscle. Cardiac muscle is only found in the walls of the heart. It's striated, which you can't really see the striations as well in this picture as you could on the previous page. We'll talk more about what we call intercalated discs, which are these bands that you can see kind of periodically in the picture. When we talk about the heart, we'll talk about what the function of those and why the cardiac muscle tissue has them. The action of cardiac muscle tissue is strictly involuntary, and we've learned about that before. It is not consciously controlled, but something unique about cardiac muscle tissue is that it creates its own nerve signal in a patch of tissue called its pacemaker, the sinoatrial node. This built-in ability of our heart to initiate its own contraction is called autorhythmicity, auto being self, so that's where that term comes from. But as we just recently talked about, things like the sympathetic parasympathetic nervous system, talked about hormones the other day, like epinephrine, also known as adrenaline, how that's going to speed up heart rate and make the heart beat faster and harder. Okay. Hormones can adjust how often that pacemaker fires. So the actual signal originates in the heart, and if the heart was just left alone, the heart rate would never change. It would be a constant, something like 100 beats per minute. But obviously, it's never going to just stay static, because just like any other controlled condition, it's very dynamic. And this changes due to outside activity from things like the endocrine system, neurotransmitters from the sympathetic, parasympathetic systems, so on and so forth, which we know cardiac is one of the things regulated by our autonomic nervous system. Another thing regulated by the autonomic nervous system is smooth muscle, as it states on this page. Smooth muscle gets its name because it lacks the striations that we see in the other two types of tissue. We find smooth muscle in the walls of hollow internal structures, like our blood vessels, our airways, many other organs, stomach, intestines, bladder, etc. Uninucleated with the nucleus located within the muscle cells, usually is involuntary, and a key feature is that although they're relatively slow moving, they are fatigue resistant, which means that they're less likely to get tired and need a break, like our skeletal muscles do in situations where we overwork them. We'll talk about fatigue with skeletal muscles later in the lecture. I don't know if we'll get to that point today or not. I doubt it, but I've got a pretty fun video to show on that when we do. All right, before we move on from cardiac and smooth muscle, which we're just about ready to do, I want to talk about the properties that those three tissues all share and allow them to function in the ways that they do. So I'll give you a second to write, and then I'll talk. Okay, our first property, the property that we should have a very good understanding of at this point in time, because we very recently talked about the nervous system. We spent several weeks talking about the nervous system and how neurons are excitable cells, so are muscle cells. Excitability refers to the ability to respond to a stimulus or stimuli, plural, by producing electrical signals known as action potentials. So that means muscles, and this is all three types, can become excited, in other words, create action potentials. Okay. Two types of stimuli signal action potentials in muscle tissues. I was just talking about the pacemaker of the heart, how it creates its own signal. We call that autorhythmicity. Whereas what we'll talk about more so in this lecture is how chemicals can stimulate an action potential in muscle fibers, like neurotransmitters. We'll talk a lot about acetylcholine actually in this lecture. Other chemicals could be hormones distributed by the blood or even local changes in pH. Now, all three types of muscle, although different, respond to an action potential the same way. When they generate an action potential, they contract, and that's what contractility, that property is. Literally, the word contract is in it, the ability to contract forcefully when stimulated. And when a muscle contracts, it generates tension while pulling on its attachment points. If this tension is enough to overcome the resistance of the object to be moved, our muscle shortens and movement occurs, which is all just like physics related stuff. The next two properties, I kind of talk about them together. It's a set of properties that other tissues have, like our lungs, for example, or skin. Think of those properties like what a rubber band can do. Rubber bands can stretch, which muscles can do, and that's what extensibility is. I think when I say that, I tell students think extend to lengthen. Now, it says without being damaged. Obviously, that's going to be within limits. You know, we all probably pulled a muscle or experienced, you know, some type of muscle injury because, you know, skeletal muscles, heart muscle, stomach muscle, muscles and other organs as well. Really, I mean, it's hard to stretch them to that point a lot of times, but it is possible. Okay? Examples there, stretching the heart when it fills with blood, stretching the stomach when it fills with food. When you let go of a stretched out rubber band, it snaps back. Okay? If you like squeeze a rubber band down and you roll it into like a smaller ball and you let go, it's going to snap back. Okay? That's what elasticity is, or sometimes you'll see this listed as elastic recoil. The ability of muscle tissue to return to its original length and shape, either after being contracted or extended. So, very important properties all three muscle types exhibit. Okay, that's it for now anyway. We'll talk more about cardiac muscle and smooth muscle later on in the semester. But from here on out, it's strictly all about skeletal muscle and how they work. First thing we are going to do before we really start talking about function and how things work is just going to give you a brief introduction into the makeup of skeletal muscles, because as I've talked to you guys about this before, structure or anatomy affects functional physiology. So let's just dive in and talk about the makeup of skeletal muscles. Most important components of a skeletal muscle are the actual muscle fibers. Fibers are just a fancy way to describe muscle cells. Okay, the diameter of a mature skeletal muscle fiber ranges from about 10 to 100 microns. What's cool is the number of skeletal muscle fibers that we have is set before we're born. So when our muscles grow, which they obviously do from when we are a baby to when we are an adult, you know, the guy on the front page had gigantic muscles. His grew tremendously, and that's going to occur due to a growth in size of the fibers, not a growth due to an increase in cell number. Most tissues, like bones, for example, grow because cells go through cell division. When our muscles grow, the cells just get bigger. They increase in width. Okay? We call that property hypertrophy. An enlargement of existing muscle fibers. The opposite of that is atrophy, which happens like when muscles shrink up as we get older, or if you don't use them. If you've ever spent time in a cast, I spent time with a cast on my left arm for like 6 months. And when I took it off, my left arm looked like, skin and bones, as they say. Real skinny because the muscles had, atrophied tremendously. But, a couple weeks, months went by, and I started using my forearm again, and, that muscle came back pretty quickly. Hypertrophy is stimulated by some hormones that we talked about the other day. Growth hormone, stimulated or released, I should say, by the anterior pituitary. Testosterone released by the testes in men, which men produce, and women don't produce that type of testosterone. They produce something else, but is one of the big reasons why men typically have bigger muscles than women. I say typically because it's not always the case. Growth hormone, we start releasing a lot of that, and testosterone as men when we hit puberty, which is where that big growth spurt comes from. Same thing for women with their growth hormone when they hit puberty. Okay, as skeletal muscle fibers mature from embryonic stem cells, many of those stem cells, called myoblasts, fuse together, which is why those cells, as a mature muscle fiber, end up multinucleated, like we show here on the next page. I'm going to bounce back and forth between these two pages. But here at the top, we show these embryonic stem cells called myoblasts coming together, kind of forming a immature muscle fiber, as it shows here. If you look hard enough in the illustration, it shows multiple nuclei. Those nuclei that are originated from each of those cells are still retained. We also have, and it doesn't talk about them on the previous page, but what we call satellite cells, which are cells that also influence the number of nuclei in a muscle fiber. Here's what a muscle fiber looks like. You can see on the outside, I talked about how the multiple nuclei are pressed up against the plasma membrane. And let's talk about the plasma membrane, what we call the sarcolemma. What you're going to see here on this page is that the organelles that we learned about a few weeks ago at the beginning of the semester when I introduced cells are the same organelles that you find in muscles, except some of those organelles, like the plasma membrane, have a different name. For whatever reason, they give some of these same organelles, like the plasma membrane, a just unique name when it comes to muscle fibers. It's the same organelle, functions the same way as it does in other cells, it just has a different name. Probably just, I guess, to confuse anatomy and physiology students. Sarcolemma is just a fancy term for the plasma membrane of a muscle cell. Like that of neurons, it maintains a resting membrane potential, meaning that it has a certain number that it rests at, and if stimulated, it can depolarize, and if enough ions flow, it can hit threshold, just like a neuron, which results in an action potential that will then propagate throughout the muscle fiber. Now, when I say muscle fiber and propagation of that action potential, what we want to have happen is the action potential to spread from the sarcolemma, that blue sheet that's on the outside, to the very internal core of our muscle fiber. There are these extensions that project inward from the muscle fiber, from the outside, I should say, from the sarcolemma. These extensions, or invaginations really, are called transverse tubules, or T-tubes for short. So their job is just to conduct nerve impulses from the sarcolemma down into the cell to ensure that an AP, action potential, excites all parts of the muscle fiber essentially at the same instant. Okay. Directly surrounding a T-tubule and throughout a lot of the interior of a muscle fiber is this kind of gold-colored, fluid-filled tubular organelle. I'll just say it that way. It's kind of hard to explain. That's called, I guess they call it a membranous sac, the sarcoplasmic reticulum, SR for short, which encircles each myofibril. I'll talk about what a myofibril here is in a second. This is essentially the exact same thing as the smooth endoplasmic reticulum was that we learned about in non-muscular cells. Okay. At the site where you have a T-tube, on both sides of it, there are fattened or dilated sacs of the SR called terminal cisterna. And since there are three things involved, we call that set of three things a triad. The T-tube on the inside and the terminal cisterns on both sides. Okay. Now, what the function of the SR is to store calcium, which is important, but more importantly, releasing calcium to trigger a muscle contraction. Okay. We release calcium, and we're going to talk a lot about this here when we get to talking about muscle contractions. Whenever an action potential spreads inward down that T-tube, we have what are called calcium release channels that open up, releasing calcium into the sarcoplasm. The sarcoplasm is just the fancy term for the cytoplasm of a muscle fiber. Okay. I'll get to that here in a minute. There's also within the SR, aside from those calcium channels, calcium pumps. Calcium pumps that pump calcium back into the SR from the sarcoplasm, creating a very low concentration of calcium in the sarcoplasm compared to the SR. And as you guys hopefully remember, anytime we talk about pumps, there's going to be a need for ATP to be involved because we're talking about primary active transport. Let's talk about the sarcoplasm. This is the fancy word, once again, for the cytoplasm of a muscle fiber, where we have a substantial amount of stored glycogen in structures called glycosomes. We learned about glycogen early in the semester, and we talked about that a lot, actually, in the last lecture. We have glycogen, which is just a fancy way to describe a bunch of glucose bonded together, which we can break down the glycogen to release the glucose so that we can then go through glycolysis to make or synthesize ATP for the energy that our muscle contractions require. Talk a lot about that here in a bit. There's also a red colored protein called myoglobin, which binds oxygen molecules so oxygen can be released for aerobic ATP production. Myoglobin is very similar to a kind of more well-known chemical in our body, hemoglobin. Hemoglobin we'll talk about when we get into our blood lecture, which is the second lecture of this unit. Hemoglobin is what causes our red blood cells to look red, and therefore is what causes our blood to appear red. Myoglobin actually causes any fluid that would be in your muscles to appear red, which includes the fluid that is found when you cook a steak to like just medium rare. A lot of people are kind of surprised to find out that a medium rare steak is not a bloody steak like people seem to think it is, because when a cow is butchered, the blood is drained. There should not be any blood left in the protein, which is the which is what our steaks are. It's just strictly protein, muscle. It's just basically watery fluid that's red in color until you cook it completely through, and then it changes that myoglobin and ends up appearing more that kind of dull gray, much less tasty version of the steak than the medium rare form. Whether you agree with me or not, I guess that is beside the point. All right, let's talk about something very, very large and prominent inside our muscle fibers that I mentioned just briefly on that previous page, what we call myofibrils. Myofibrils are these tubular structures that have a contractile function inside our muscle fibers. What I'm going to do, and I do the same thing in anatomy, is just kind of give you a brief explanation on the way that our muscles are organized, which is important. Kind of keep your head straight on things and give you an understanding how amazingly we're built, but also complicated we're built within our muscles. So I just call this a flow chart. I'll start at the top with the largest structure, which would just be the entire muscle itself. If it will focus for me. Which is then subdivided into smaller structures called fascicles. Fascicles are just a bundle of fibers, which means that our next level of organization are fibers, Fibers, which we just learned, are the fancy way to describe our cells. And inside of those cells are these myofibrils that I was just talking a little bit about. I basically just described them as contractile organelles inside our muscle fibers. And myofibrils are long and tubular, but then are subdivided into segments called sarcomeres, which we'll talk more about on the next slide from the one that we were just currently on. Sarcomeres are just the functional units of a myofibril. And those are made up of or found inside of sarcomeres structures called filaments, which are sometimes described as myofilaments. And the myofilaments or filaments are what participate in the contraction, ultimately shortening a sarcomere or sarcomeres, which cause the myofibrils to shorten, which cause the fibers to shorten, causing the fascicles to shorten, which means what happens to the entire muscle. Not a trick question. They shorten as well, or it shortens as well. Looking at our figure here, we show that singular muscle fiber, which is what this is, contains a whole bunch of these parallel to one another myofibrils. If we pull one out and kind of open it up, we see that there are bracketed off regions here. We show one of them, a sarcomere, is a functional unit. Inside of the sarcomere are the thick and thin filaments, which overlap one another in some segments, but don't overlap each other in all the segments. Obviously, that's something we'll talk more about here on the next slide. But it's this makeup of the sarcomeres and the thick and thin filaments that ultimately cause our skeletal muscle fibers to appear striated. We see the same pretty much same, makeup to cardiac muscle. We see myofibrils and T-tubes and sarcoplasmic reticulum and all that stuff. So we'll get into that when we talk about the heart, which will be unit number 5. Okay, let's talk about the sarcomeres and filaments a little bit more in detail on this slide. Okay, reading through. Within myofibrils are smaller structures called filaments, which there are 2 majors. There's actually a third that I'll talk about as well. The thin filament and the thick filament are the 2 majors, literally named because one's thicker than the other. The thin filament's about 8 nanometers in diameter, one's 2 microns in length. Thick filament's twice as thick, 16 nanometers in diameter, and about 1 to 2 microns in length as well. Both of these are involved in the contractile process, as I mentioned. Filaments inside of a myofibril do not extend the entire length of a muscle fiber. Instead, they are arranged in compartments called sarcomeres, which are the basic units of a myofibril, which we are showing off here on this next slide, and the kind of anatomical makeup of a singular sarcomere. We have kind of more of a 3-dimensional version of the figure at the top and a more 2-dimensional figure down here at the bottom to show the parts of a sarcomere. I'll give you a quick rundown on the parts, starting with what we call Z-discs, which there are 2 per sarcomere. They are the zigzaggedy looking things on the edges of a sarcomere and therefore are the borders of a sarcomere. Z-discs are the dense protein material that serves as a place of attachment for our thin filaments, which we show down here at the bottom with the yellow lines up here with the kind of skinnier red lines. Notice the thin filaments attached directly to the Z discs. Now, the Z discs are also important not only for, well, how do I say this? The Z discs are a component or found within a segment of a sarcomere called an I band. Okay. An I band is found on both sides of a sarcomere. In fact, an I band actually spans across two sarcomeres because there'd be another sarcomere going in this direction and another one in this direction. So a Z disc can be described as the middle part of an I band. Now, I was talking about striations earlier and how our skeletal muscle has these light, dark, repeated stripes. The light striations that are seen in skeletal muscle are due to the I band. Now, an I band is strictly where you find only thin filaments, no thick filaments, okay, which are what we're showing here in red or up here with the darker red bands. The A band is going to span from one side of a thick filament to the other side of the thick filaments. Okay. And an A band is what appears dark with regards to striations. So this is where the light, dark, light, there'd be another A band, so that'd be dark, light, dark, light, dark. That's where the repeated arrangement of light, dark striations comes from. It's kind of handy. I bands, I don't know if they named them this way, have an I, so does the word light. Dark has a letter A in it, as does A band. Now you can subdivide the A band down into three zones. Two zones of what we call overlap, which is called zones of overlap because this is where the thin and the thick filaments are overlapping one another. See those on both sides. And then the H zone is the segment of the sarcomere where there's only the thick filament. There is no thin filament within the H zone. At the center of, or the middle part of a sarcomere is what we call the M line, which is where you find supporting proteins holding those thick filaments together. And it's at the center of the H zone. Now, the pattern of the thin and thick filament overlap, consisting of zones and bands, creates the striations that can be seen in single myofibrils and in whole muscle fibers, as I mentioned earlier. The amount of overlap of the thick and thin filaments is dependent upon whether the muscle's in a contracted, relaxed, or stretched state, which is illustrated with this figure that I have a couple slides ahead, where we show two sarcomeres highlighting the H zone in green, the I band in blue, the A band in pink. Notice what happens as we go from a relaxed state where the muscle and the sarcomeres are stretched out to where it shortens partially, when partially contracted, and then completely maximally contracted. The H zone, where there's only the thick filament, goes away as we go from relaxed to maximally contracted, as does the I band. What's going to take place when a sarcomere shortens is the Z discs are going to get shoved towards the M line because these thick filaments are going to grab and pull on a thin filament, pulling the thin filaments towards the M line, which means the zone of overlap is going to grow, the H zone is going to shrink, and the I band is going to shrink. What we show in this figure, and I really love that they do so, is that the A band does not change in shape or length. And that's because the distance from here to here, the A band is not going to get shorter. The thin filaments don't change their length. What happens is the amount of overlap changes. Literally, the theory to describe a muscle contraction is called the sliding filament theory, because the filaments slide over one another, ultimately shortening the sarcomere, which ultimately shortens the entire muscle. Now, I include this portion of the figure, and I really like this portion of the figure because it shows, and we have to think about it this way, in a three-dimensional sense. Because muscles are three-dimensional, and all those different things that I wrote out on that sheet of paper are three-dimensional, which includes the components of a myofibril and the sarcomeres with the thick and thin filaments. Each of these dark red big circles represents a thick filament, and all the smaller, tinier dots represent a thin filament, which, if you look at all these except the ones on the outside, there are 6 thin filaments that surround a centralized thick filament. Every 60 degrees, you find a thin filament surrounding the internal thick filament. Okay. And now we haven't talked about the thick filament yet, but what's really cool is the thick filament is made up of proteins. So if I pretended like this is a dot like that they had on that page, there are little extensions that come off in all different directions. Those are the myosin heads that I'll talk about. And they're going to be able to grab and pull on the thin filaments in all of those 60-degree different directions. So it's really cool. Let's talk about myosin and actin, as I just mentioned the word myosin. Those are the two what we call contractile proteins that are found inside our myofibrils. If I go back to this figure, in parentheses behind, and I know it's very small on your page, but that's okay. Behind the thick filament in parentheses, it says the word myosin. Behind the thin filament, it says actin. Okay. These proteins that are part of the thick filament are made up of a protein called myosin, which functions as a motor protein in all 3 types of muscle tissue. In this case, we're focusing on skeletal. Myosin is a protein that will push or pull various cellular structures to achieve movement by converting the chemical energy in ATP to mechanical energy in motion. Bouncing back to this page, not to talk so much about this figure, but to talk about these figures on the left. Starting with this figure at the top, which is labeled as a singular myosin molecule, at least portion of it. I always tell students, and it's kind of what the textbook describes it as, a set of 2 golf clubs where they're twisted around each other in the shaft region, and then the heads are at the distal end, like we show here. Now, unlike a golf club, which is very rigid, these heads have the ability to swivel, where they can change the position of the head by way of moving at what it's shown here as its hinge. I always use my wrists as kind of an easy demonstration of them. You know, if I put my hands in a fist, they kind of look like those myosin heads. Okay. A myosin head can go from an extended, or I will show you, it's described as a cocked position a lot of times, with the power of ATP to a flexed position. Now, why that's important? Because when it goes from this position to this position, it's doing so while it's pulling on the thin filament. Okay. There are 2 major binding sites on each head of a myosin molecule. The ATP binding site, where ATP binds to it to energize it into its cocked or extended position. And then the actin binding site, where that myosin head can attach to an actin molecule. Okay. Now this is just one myosin molecule. There are approximately 300 of those that make up a thick filament. Some are running in one direction towards a Z-disc on one side. Some are running the other direction towards Z-disc on the other side. And as we learned, the M line is made up of protein to kind of support those myosin molecules running in the opposite directions. Myosin is the only protein that makes up the thick filament. A thin filament, as it said in parentheses, is made up of actin, but there's also two other proteins that I'll talk about here in a second that help to form the thin filament. Let's talk just about actin first. Actin is our other contractile protein, the main component of the thin filament. There's actually kind of two types of actin. What we call G-actin is where they are individually kind of like beads to a beaded necklace. Each individual bead is a G-actin, and each of them has a black dot representing its myosin binding site. When stranded together, they form a two-stranded helix, which is what the F-actin is, where we can see all those myosin binding sites are kind of in the same position, where the myosin head can grab and hold on and eventually pull on the actin, thus pulling the thin filament towards the M line. Okay. Actin, as I said earlier, is anchored to the Z discs. The other two components of the thin filament are the regulatory proteins tropomyosin and troponin, which help turn on and off the contraction process. And this is where calcium comes into play that I mentioned earlier. All right. Let's talk about tropomyosin and troponin. I'll go back to that slide if you didn't get it all written out. Tropomyosin is a kind of rope-like protein that when a muscle is in a relaxed state, covers the myosin binding site of each of those actins. But when calcium is released, as we mentioned earlier, calcium is very important, calcium binds to the other regulatory protein called troponin, which is going to cause, I'll show you a picture of it. Right here, calcium is binding to troponin, and look what happens to the tropomyosin. It moves out of the way of those binding sites, which then enables, if the myosin is ready, to bond to and form what's called a cross bridge. I'm getting ahead of myself because this is what we'll talk about in a little bit. But when a cross bridge forms, then the myosin can grab and pull on the thin filament and ultimately end up doing what we showed over here in this set of pictures. Okay, there's a third set of proteins inside of our muscle fibers I want to talk about, what we call structural proteins, which help keep things put together correctly, keep the thick and thin filaments in proper alignment, give the myofibril the elasticity and extensibility properties we talked about, as well as linking myofibrils to the sarcolemma and extracellular matrix. If I go back to our illustration of the sarcomere, I did not talk about this thing that kind of looks like a coil, what we call titin. Titin is the third most plentiful protein in our skeletal muscle, coming in after actin and myosin. Gets its name because it's super big, relatively large sized, and it acts to stabilize the position of each myosin by spinning from Z discs to the M line. Very important to know what really titin accounts for is the ability to extend and recoil or snap back, as I kind of described it as, like a rubber band of our myofibrils, therefore our entire muscle. Other Structural proteins include dystrophin, which links thin filaments of the sarcomere to integral membrane proteins in the sarcolemma. Anybody ever heard of muscular dystrophy before? Muscular dystrophy, unfortunately, very unpleasant disease for people to suffer from. The most common form of muscular dystrophy, I think it's Duchenne's, how you pronounce it, muscular dystrophy, is a result of essentially not producing the correct or a valid form of dystrophin, and it, over time, essentially just causes the muscles to stop working correctly and ends up paralyzing people. No good way to prevent it either, so really, really sucky. Nebulin wraps around the entire length of each thin filament, helping to anchor them to the Z discs. There's a couple others that I didn't list that are structural, so just focus on these three. Okay, now the fun really begins where we dive in on muscle contractions, learning about all that wonderful stuff. Now, what I've included on a lot of these slides for the next, I don't know, 10 or 15 slides are various pictures to help kind of explain and illustrate what's taking place. I know typically I don't ask you guys to know many pictures in this class. That's more of an anatomy thing. But this is kind of a set of images that I want you to know. Where, you know, obviously at the bottom it tells you what's happening, but what I would do is I would, you know, erase that off, cut that out, and then I'd put this picture on the exam, and, you know, it'd be like a multiple choice style question where I'd say, okay, explain what this picture is illustrating. Or pick out the choice below that's explaining it. Okay. Now, if I were to follow the textbook as far as order goes, like I do in anatomy, we would have talked about muscles before we have talked about the nervous system, okay, which I don't like to do in this class because we, at this point, have a very good idea of what takes place with regards to action potentials. And an action potential, as we've learned, is essential for a muscle to contract. Now, unlike the heart, which creates its own action potentials, our skeletal muscle relies on action potentials coming from somatic motor neurons. So a skeletal muscle fiber will remain at rest until it's stimulated by a somatic motor neuron. Okay. And when stimulated by an action potential, the motor neuron releases acetylcholine into its synaptic cleft, where it then crosses the cleft and binds to receptors on the other side. We all know all that wonderful stuff that is involved with synaptic transmission, where calcium enters the bulb, synaptic vesicles move towards the cleft, exocytosis occurs, in this case, acetylcholine is released. Now, on the opposite side of the cleft are receptors on what's called a motor end plate, which is something that was actually labeled in that picture, or pictures, because it's kind of two pictures. The motor end plate is just the muscle membrane on the other side of the cleft, where you have an insane number of receptors for acetylcholine binding specifically to acetylcholine, ACH. Okay. What's cool about the motor end plate, if you look hard enough in the illustration, is that this is the sarcolemma. So this is the plasma membrane of our muscle fiber. The motor end plate is invaginated. It's got these inward extensions, kind of like what we talked about with cells that had microvilli. That increases surface area, which means, and you can see it right here, each of these little purple boxes is one of these acetylcholine receptors. So by being invaginated, you have a lot more of those receptors, which means there's more areas for acetylcholine to bind, right? Okay. Well, we're zooming in here on this page to one of those acetylcholine receptors, where acetylcholine binds to that receptor, and it's like a key unlocking a door, which opens the door up, allowing sodium channels to open, allowing sodium in, which creates a local potential on the muscle membrane. Absolutely, yes. All the pictures, unless I tell you otherwise, for the next several slides. Not all the pictures in the lecture, but when it comes to this stuff. Now, if you think back just a few weeks ago, we learned that if sodium rushes into a cell, if enough sodium rushes in, we can hit what? Threshold, right? And what happens when we hit threshold? We create an action potential, right? So you're starting to see why I like to talk about action potentials before we talk about this stuff. Because look what we're going to talk about on this page. Muscle fiber excitation is the process in which action potentials in the nerve fiber lead to action potentials in the muscle fiber. So an action potential in our neuron triggers an action potential in our muscle fiber, our muscle cell. Okay. Those green blobs here in our picture are acetylcholine molecules in the cleft binding to those receptors that we just talked about, creating a local potential, local or greater potential, if strong enough, hits us to threshold. And if we hit threshold, we still see the same channels that we learned about previously. Voltage-gated sodium channels open up, sodium rushes in, we rapidly depolarize, and then those close, and voltage-gated potassium channels allow the potassium out, causing us to repolarize, returning back to resting membrane potential. Now with neurons, we saw an action potential traveling down the length of the axon, right? Down the long wired part of the neuron. In muscle fibers, the action potential is going to originate, maybe we can just say right here. That action potential is going to spread throughout the entirety of the plasma membrane, the sarcolemma. But remember, we've also got these structures that projected inward called T-tubes. And I'll get to that here in just a second. This figure, actually it's 3 figures, is actually from your textbook. These figures that we've seen so far is from a different textbook that I used to teach with, and I've just kept the pictures because I like them a little bit better because they go kind of step by step. Your book kind of gives you more encompassing pictures. But I still like this picture or these pictures to show some of those things I've talked about already. Here's our muscle fiber. On the outside is its sarcolemma. Here's one of those synaptic end bulbs. We zoom in on it. We show the motor end plate across. We zoom in on it. We see the acetylcholine coming across, binding to those receptors, allowing sodium in. And if enough sodium rushes in, we create an action potential. Like I said, that's going to spread throughout the entirety of the sarcolemma, but also down into the middle of the fiber by way of those T-tubules, short for transverse tubes or transverse tubules, which brings us to this slide. Okay, and our picture over here on the left side, these yellow arrows, just think of those as the action potential spreading from that motor end plate across the plasma membrane, the sarcolemma, but more importantly, into the center of the muscle fiber, thanks to those transverse tubules, T-tubes for short. And remember, on both sides of a T-tube are fattened portions of the sarcoplasmic reticulum, the SR, called terminal cisterna. That is where you have channels, literally called calcium release channels, that tell you exactly what they do. Release calcium. Calcium that's dumped out of the SR, into the sarcoplasm, okay, where that calcium is going to flood and find the troponin and tropomyosin stuff that we were talking about just a little bit ago. Okay, so reading through, as the stimulus travels across the sarcolemma, it will travel down into the muscle through T-tubules. Stimulus causes the release of calcium from the sarcoplasmic reticulum, SR for short. Calcium concentration in the cytosol is typically very low, but when the action potential reaches the T-tubes, it is released in large amounts out of calcium release channels. Now, we haven't contracted yet, but we're getting close. Excitation contraction coupling, which you might put in parentheses ECC for short, refers to the events that link action potentials on the sarcolemma to activation of the myofilaments. So that's the thick and thin filament stuff we talked about, thereby preparing them to contract. We just said calcium flows out from the terminal cisterna, the SR, through the calcium release channels. Then that calcium binds to troponin, like we show here on this page. This figure I kind of already talked about over here is what happens next, where the calcium binding to troponin causes a shift in tropomyosin, exposing, and it says here, active sites, and it shows them labeled here as active sites. You might put in parentheses, active sites are the same thing as myosin binding sites. Active sites equal myosin binding sites. A site where a myosin head can attach and grab hold of an actin. But myosin, in this case, right here, has got its heads flexed. And they can't grab and pull on the actin until they are in extended position, like they are down here in step number 10, as it says, which requires the hydrolysis of ATP to ADP MPI, which activates it, and I said earlier we use the term cocked or cocking it into its extended position. Okay. So calcium then binds with the troponin complex, causing the tropomyosin to rotate and open up the myosin binding sites. Myosin heads then can attach and go through a contraction cycle, which we'll get to here in a second, which means that they form a cross bridge and then end up pulling the thin filaments towards the M line, as I showed. Okay. This is going to continue to happen as long as calcium is prevalent in the sarcoplasm and as long as we have enough energy to keep causing the myosin heads to extend and then break them apart, so that break them from the actin so we can form a new cross bridge. Once again, a cross bridge, I've used that term a lot. It's just the bond between actin and myosin. Muscular contraction is the step in which the muscle fiber develops tension and may shorten. Model, I described this earlier, or said this earlier, describing muscle contraction is called the sliding filament theory, which states that actin and myosin myofilaments do not become shorter when they contract. Instead, the actin slides over the myosin, causing the muscle fiber to shorten. The power stroke, sometimes you'll list it as, or see it listed as a working stroke, is where we finally grab and pull and slide that thin filament towards the M line, like we're showing going from 11 where it's attached to the flex position as it pulls the thin filament over the thick. Okay. Now at this point, we've done all we can with this thick filament in this state. So what we're going to end up having to do is release that connection, that cross bridge between the two, so that we can go back to a cocked position, which is one of the steps of our cross bridge cycle that I'm going to discuss on this page. Cross bridge cycle, also known as contraction cycle, is a 4-step process that we show here on this page. Now, if we, actually, I'll just wait and say that here in a second. Let's just talk about the contraction or cross bridge cycle. So in order for muscle shortening to take place, actin and myosin must continually cycle through the contraction cycle. In the resting state, myosin filament is unattached to actin, and ATP is already separated into ADP and PI. Okay. But as we know, when calcium binds to troponin, tropomyosin shifts, exposing those binding sites, which then prepares us to form a cross bridge, okay, which I'm getting from 1 to 2 here. So let me stop right back here at number 1. Shown right here, ATP hydrolysis is the name of this step, where the myosin head takes that ATP, hydrolyzes means to break it apart, forming ADP and PI, which takes it from that flex position to the cocked position. It energizes it, it extends it, whatever you want to describe it as, so that it's ready to then, if calcium causes a shift, to form a cross bridge, which is our second step attachment, where the myosin head attaches to the thin filament, to the actin, forming a cross bridge. We then go through the power or working stroke. As I said, it's also sometimes described, where the ADP and PI molecule is released. Myosin head then pulls the thin filament and slides it over it like we show right here. Once again, in this state with the head flexed, the myosin is no longer able to do any good, so it must break free. We call this detachment, which requires ATP. ATP that then binds to the myosin head, breaking it, and then we use that ATP to hydrolyze it and extend it and start it all over again. This is going to continuously happen as long as 2 things are prominent in our muscle fibers. Calcium, so that we have a shift in the tropomyosin. ATP, so that we can break the cross bridge and extend or cock the head. If we run out of either of those, muscle contraction cannot occur. Now, we'll talk about muscle relaxation here in a second. Anybody ever heard of rigor mortis before? What is it? Tightening of the body, and that's because the body, when we die, after death, we release calcium from the SR, causing this to occur repeatedly until we use up all of our ATP. And then we get stuck right here, meaning we can't break the cross bridges. And if the cross bridges can't be broken, then the muscle can't relax. And so a stiff, as they are described, is a dead body where the muscles are basically kind of frozen in a contraction process. And it lasts for a few hours, and then it kind of goes away. But we've ever, seen a dead body, it can be kind of crazy in the positions that they get left in because of rigor mortis. Okay, let's talk muscle relaxation. What's that? Acetylcholine esterase. Rolls right off the tongue, doesn't it?
Yeah.
Or you could abbreviate ACHE, which is much easier. Okay, now, how did we begin our muscle contraction? Well, we trace it all the way back to our motor neuron creating an action potential and propagating it down to the synaptic end bulb. Therefore, we can trace muscle relaxation back to the same thing. This time, it's the no longer action potential propagating down the muscle or the neuron to the synaptic end bulb. Okay. When a nerve signal stops traveling to the synaptic end bulb, we stop releasing acetylcholine into the cleft. No acetylcholine in the cleft means no binding to the receptors. Any remaining acetylcholine is going to be removed from those receptors thanks to that enzyme acetylcholine esterase. Now, the figure that I've got here actually shows this taking place kind of as towards the beginning. And that's because it's not like we only have acetylcholine esterase active at the end. It's going to be continuously active. The thing is, if we still have an action potential, we still are releasing acetylcholine. So we're breaking it down, but we're also releasing it in new acetylcholine. So its effect's really not visible till the very end, which is why this has got it listed as step 15. Now, what's cool is the acetylcholine actually gets broken down, but it's recycled into its parts so it can be then used to be made again, essentially. No acetylcholine means no action potential in our muscle fiber. So we no longer see our yellow arrows traveling down the T-tubes, which means no calcium is going to be released from the sarcoplasmic reticulum. And the calcium that's in the cytoplasm gets pulled out of there and put back where it came from. Back into the sarcoplasmic reticulum by way of calcium active transport pumps, which sometimes you'll see those listed as calcium ATPase pumps, which tells you exactly what's involved. ATP. ATP is not only needed for muscle contraction, it's also needed for muscle relaxation. Because remember, active transport requires ATP because we're pumping against the gradient, right? From low to high instead of from high to low. Okay. We're putting the calcium back in the SR. We also have within the SR calsequestrin, which is a protein that allows and helps with storing extra calcium in the SR. So it helps build it up in there. No more calcium on the inside of the muscle fiber means no more calcium binding to troponin, which means we're going to shift the tropomyosin back over those binding sites, active sites as it's listed down here, which means that, and it doesn't show it, but if this myosin was in a position to bind to actin, it couldn't, because that is blocked. And so removal of calcium from the sarcoplasm causes a troponin-tropomyosin complex to shift over and cover the active sites. Active sites, same thing as myosin binding sites for myosin attachment, stopping the production or maintenance of tension. Muscle fiber then can return to its original length due to its recoil of the series elastic components. That's all about the tighten, along with the fact that a lot of times we're relaxing muscles, but at the same time we're also contracting antagonistic muscles. Like, you know, when we extend our elbow, we're contracting our triceps, which causes our biceps to return to its resting position because those are antagonistic pairs. Here's the illustration. Like I said, your textbook kind of gives you just more of an encompassing overall view of things instead of step by step. Not that this is a bad picture by any means, but I like to go through things kind of individually instead of just all at once. Some of it's kind of cut off. Oh, sorry, I hate when this happens. So starting at the top, nerve signal travels down our down to the synaptic end bowl above our somatic motor neurons, triggering the release of acetylcholine, causing them to bind to receptors on the motor end plate, which causes an action potential if we have enough sodium rushing in. Action potential travels down into the T-tubes, triggering the release of calcium from calcium release channels. Calcium is then going to flood into the sarcoplasm where it can then find troponin, bind to it, shift the tropomyosin, causing us to form cross bridges, and thus, if we have enough calcium and ATP present, go through the contraction process, causing our sarcomere to shorten like we show here. This is going to continue, like I said, until we stop sending the signal down, which means we stop releasing calcium. Any calcium that's still left over gets put back into the SR by way of the calcium active transport pumps, or calcium ATPase pumps as they're listed here, which causes the shift to occur right in the opposite direction. We cover the binding sites, and the muscle can then therefore relax. To summarize, I've included a step-by-step, kind of very briefly, but very nicely list of the 11 steps here, which is something that I'm not gonna ask you to reiterate and write down on the exam step by step, but is absolutely something that you should be able to know. Kind of like what I told you guys with action potential steps, synaptic transmission steps. Take out a blank piece of paper once you feel like you know this stuff and test yourself out. See if you can write out the 11 steps from 1 to 11. If you can, fantastic. If you can't, study some more. Because that's going to be a lot of questions on your exam next week. Okay, to kind of help us, I've got a couple videos here.
The process of excitation, contraction, coupling consists of all those processes involved between the time of generation of the action potential on the muscle sarcolemma and the actual contraction. As the action potential reaches the neuromuscular junction, vesicles containing acetylcholine fuse with the membrane of the nerve terminal, and acetylcholine is released into the cleft. Acetylcholine molecules rapidly diffuse across the cleft and bind to the acetylcholine receptors. Binding of acetylcholine to receptors immediately opens ion channels, allowing sodium ions to rush into the muscle fiber and potassium ions to diffuse out. Acetylcholine is then split to acetate and choline by cholinesterase, resulting.
All right, they called it cholinesterase. We called it acetylcholinesterase. Same thing. Like I said, it's taking place all the time. But as long as we're still continually releasing that acetylcholine, it's not really going to produce an effect until the very end.
In inactivation and closure of the channel. The initial flux of ions due to opening of the channels brings the membrane potential to threshold, causing the voltage-gated sodium channels in the vicinity to open. The resulting action potential is propagated over the entire surface of the muscle and also courses into the T-tubule system. As the action potential encounters DHP receptors in the T tubules, it causes opening of the ryanodine calcium channels of the laterals.
Okay, ryanodine channels, that's just a fancier way, and I'm not going to want you to know it that way. This is just a calcium release channel because it releases calcium. Simple, easy way to remember.
Sacs, thus allowing calcium ions to diffuse out of the lateral sacs and into the region of the myofibrils. Calcium then binds to troponin, resulting in movement of the tropomyosin-troponin complex away from myosin binding sites on actin. The contraction process continues as long as action potentials continue to reach the neuromuscular junction, thereby keeping the concentration of calcium elevated around the myofibrils. To end the contraction, calcium must be actively transferred by the calcium pump, calcium ATPase, back into the lumen of.
Okay, calcium ATPase, like I said, calcium active transport pump, same thing. Here's ATP that's using that to pump it against its gradient from low to high.
The lateral sacs. This pump utilizes ATP as a source of energy. This process reduces the concentration of calcium in the vicinity of the myofibrils, such that calcium dissociates from troponin. Tropomyosin again covers myosin-binding sites on actin, and the muscle relaxes.
Okay, that video's okay. I also really like this one. Play it.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber,
Well, maybe we won't watch it. I don't know why it's not playing. Tell you what, I'll see if it'll load for a little bit, and then we'll try it. If not, I'll try to find a new video and show you guys next time. So that is quite a bit, right? Complicated. I would say about this point is kind of the point where, going back to the very beginning about the first 20 questions, or about half of the questions for the 40 that come from this packet come from, about the second 20 or the other 20 come from the rest of the lecture. I'll tell you what, this seems like a really good spot to stop, because I imagine your head's probably spinning as it is. So I will call it a day here. This is where we'll pick up after our exam on Thursday. If I get that video to load for next time, we can watch it then. See why it's messing up.